Dear ladies and gentlemen, I would like to welcome you to the last edition of our BioM Lunchtalk in 2024. We have decided to do it in English again to allow a broader audience to listen. Most of you know that BioM is the cluster management and cluster development company for biotech in Bavaria, providing support and services to our biotech ecosystem. My name is Andreas Berghammer, and I'm responsible for services, cooperations, and sponsoring at BioM. So, if you would like to participate as a company in an event, for example, such as BioConnect next July, please contact me and be with us. But now to today's topics, which are a little different from the previous ones. We often had short series of presentations like on cell, gene, and immune therapies, on AI, ML for biotech, or therapies based on nucleic acids. Today, we wanted to introduce a company and an organization with very special skills and expertise, which might help you on the road from R&D to clinical phases. The title of our lunch talk is therefore Advanced Biomedical Development and Clinical Trials, which I have to admit is not a very narrow title, but we come to the details. Today, we welcome representatives from HTCR-Services. I will use the German spelling, HTCR, and from Fraunhofer IES. Before we start, most of you know that as well, some remarks on the format. As always, we will first have a presentation of about 20 minutes. Afterwards, we will have a few minutes for direct questions, which we will read out anonymously from the question chat for data protection reasons. You are therefore welcome to post your questions already during the presentation. This will be followed by the second presentation, 20 minutes, again, some direct questions, and finally a round with both speakers, where you can ask questions on both topics or on overarching topics. We will start with HTCR-Services. HTCR is the acronym for Human Tissue and Cell Research. The company is a wholly owned subsidiary of HTCR Foundation with a vision of making high-quality human biospecimens available to the medical research community. Founded in 2002 as a spin-off from the University of Regensburg, HTCR-Services offers human tissue-based products and services for medical research and drug development applications. Our speaker today is Mareike Ahrends. She's project manager at HTCR. Mareike studied pharmacy in Berlin and Florida, did her PhD in Hanover in molecular medicine, and added a training recently in life science management. She joined HTCR this year. She will talk about HTCR as a partner for human tissues, cell models, and contract research. Mareike, we are looking forward to your presentation.
Yeah, thank you, Dr. Berghammer. I am really blessed to be invited here to speak here in this really interesting session. And basically, you already introduced not only me, but also the HTCR. So there might be a few things that you already know. So I must honestly say that you came up with a brilliant title for this session. So Advance Your Medical Development and Clinical Trials, because that is exactly what we do. And I'm soon going to tell you why and how. And you got the teaser already, because we are your partner for human tissue and cell models and contract research. But first, we'll have a quick look into what medical development actually means. So most of you already know that, but especially since the COVID-19 pandemic and discussions about vaccine development, it has become common knowledge that the drug development process is usually quite expensive and time-consuming. So typically it takes several years of research and development and preclinical studies in which a library of several thousands of compounds was narrowed down to just a few promising candidates, which then move into a clinical trial in humans. And it takes even more years and a lot of more money until there is enough data to actually get one of these approved and marketed. However, those clinical trials often fail to reproduce the preclinical results. And furthermore, scientists actually don't want to typically use animals as a model in research. You might know the 3R principles: to replace, reduce, and refine animal experiments. And the major argument here is that those animals that are used, or these models that are used, also other preclinical models, just do not recapitulate the human situation sufficiently. So the results are often not predictive and not translatable. Fortunately, by now, there is an entire field addressing this issue, which is translational research. And before I joined HTCR, I actually worked for a Fraunhofer Institute as well. In translational science, I had a focus on preclinical pharmacology and toxicology regarding airway diseases. And we mainly used human lung tissue as an alternative model to animal experiments to not only spare mice, but also to produce human relevant data, like more human relevant data. And this has an enormous potential, but it comes with a limitation of low accessibility, because no living healthy human would just give part of their lungs or other tissues to researchers for fun. So we could just have done so many more studies and gained wonderful results if there was only enough tissue. And there were surgeons at the university clinics in Regensburg, as we heard already, who had a quite unique approach on how to address this issue. In the year 2000, Professor Jauch founded the nonprofit foundation Human Tissue and Cell Research, the HTCR Foundation. So that again is what HTCR stands for: Human Tissue and Cell Research. Around about the same time, his colleague Professor Tassler and others founded a company which is now the HTCR-Services, to take care of all the operational tasks that this foundation has. By now, the HTCR-Services and the foundation moved to Munich. The HTCR-Services is fully owned now by the foundation and also operates the LMU Clinics Biobank. And I brought to you a kind of historic article that actually sums up all that quite accurately. It was published in the Middle Bavarian newspaper in December 2000, and it starts by stating that animal experiments could soon be obsolete. It continues that leftover tissue from surgeries, which would usually be disposed, can be stored in biobanks to be used for scientific purposes later on. However, what makes this, well, kind of complicated construct so clever and so unique is what comes next. The foundation offers more legal security and a separate ethics council to address ethical concerns and to also monitor the proposals, so the scientific projects. Upon an informed consent of the patient, they then donate their tissue to the foundation as a trustee to support scientific research in a non-profit manner. By now, we have a clinical network of currently 4 major clinics in Bavaria, and those provide us prospectively with different tissue entities and disease backgrounds. Last year, for example, the majority originated from colorectal and gastrointestinal surgeries, but also from skin, lung, and liver resections. And so once the surgeon here on the left resects the tissue from the patient and the pathologists get their necessary share for the diagnostics, we continue to process the sample in our labs. So we examine the tissue, divide it, and snap freeze a sample for long-term storage, which is the classical biobanking activity that we do. Most of that is done at our main laboratory facility at the LMU clinics. That works on a very well-established QM system and is also part of the German Biobank node. So to also include some actual numbers, you can see on the left in this diagram that the number of patients in our biobank was nicely and steadily rising over the years. By now, it contains almost 7,000 donors, which sums up to a total of around about 40,000 individual samples that can be inflamed or tumorous or metastatic tissues or diseased tissue, but also healthy tissue, like macroscopically normal, so to say, or also isolated primary cells and blood. But in addition to this tissue, we also provide clinical patient data. And this includes patient characteristics, but also medication, pathological information, pre-treatments, and certain lab markers. And to prevent the re-identification, so to protect our patients, our sample donors, this data is de-identified twice, actually. So once the biospecimens come in and once they go out. And to collect and store this data, we use the Centra X40 database by Kairos. This is a state-of-the-art tool, which is also used by many other clinics to access and evaluate biomedical patient data. The license is unfortunately quite expensive, but it's really good though. So this is the classical biobanking that we do. However, we can do much more. And additional to the collection and storage, we also provide the samples as fresh tissue. And this fresh tissue offers completely new possibilities compared to the stored or fixed tissue. And this diagram of the NC3R, that is a UK-based science organization. This sums it up quite nicely. So fixed tissue on the right is typically used for things like imaging or molecular pathology, nucleic acid recovery. So to analyze a status quo, that can be a biomarker identification or some endpoints. But fresh tissue, on the other hand, can be cultured. It reacts to a stimulus or to whatever intervention that you might want to analyze. You can have functional readouts, you can have organotypic cultures, or even include microphysiological devices to get an even more human relevant and real world-like conditions. But also if you're working with primary human tissue, you know that the results hinge on the initial tissue quality, and that is mainly viability. And the main part that influences viability is time. So not only ischemia time, so the duration in which the tissue is without nutrient supply, but also the time until you can actually start your experiment. And so we optimized our procedures in our lab facility to ensure quality throughout shipping and have those samples arrive at our partner's place on the next morning. And this way, we perform around 500 allocations of fresh tissue each year, slightly less during the COVID pandemic. Also, by the way, one allocation is not necessarily one sample, one biospecimen sample, but it can also be a set of several samples from same patient, like a set of matching disease and healthy tissue, plus isolated PBMCs and serum. And the majority of those allocations is on academic projects. You can see the blue bars to meet the foundation's guidelines. of primarily supporting non-commercial research. And besides the timing aspect, we have also optimized our reagents to increase quality. We even have our own patented cold storage solution, which is used for shipping. This is the patent certificate still hanging in our office. And we also optimize our protocols. For example, when it comes to the isolation of liver cells, we receive both hepatocytes and non-parenchymal cells in parallel. And those results can have been published just a few years ago. You can here see the different subsets of Kupffer cells, stellate and endothelial cells, and quality maintenance, not only in terms of viability and morphology, but also functionality of those hepatocytes. And to show you some more of the enormous potential of these fresh tissue allocations, I brought to you this recent publication of one of our long-term pharma industry partners who just published in quite a high-ranking journal, Nature, where we provided intestinal tissue samples of 27 donors, and they were able to generate intestinal organoids that include the naturally resident immune cells. And they showed, like those organoids showed clinically relevant functional features also. Please don't ask me any details about this, but I highly recommend you read that publication. And also I brought to us this very nice video clip of that same publication to enjoy, where you see the epithelial cells of these intestinal organoids in bluish and the tissue resident memory T cells in pink, which are wiggling their way through that. I really enjoy this video. Science can be really beautiful sometimes. So you might wonder how you can get your hands like practically on those samples. It's not too difficult. So you basically go online to HTCR-Services.com, then you're forwarded to the foundation's website where you find this application sheet. I just gotta admit that we just renewed it. So this would be the current request form. We're about to update the website, and then we can check feasibility, have our MTA sign and send you an offer. And then you fill out the project application form, request an ethics vote, and of whatever ethics board, ethics council is responsible for your case, and have it approved by the foundation also, mainly Professor Jauch, the original founder, and Mr. Tossler, and then you're good to go. Or to make it a little more simple, you can just ask me or one of my colleagues. Our job is to support you and to make sure that you get the best samples that we can possibly offer, so that you can get the best results. So this is our small but excellent team. Christina, our lab manager at the LMU site and biobank coordinator. Torsten, our lab manager at the IZB site. Peter taking care of our database. Then there is Dennis and Saskia, our lab technicians. Chao Shun and myself as project managers, and our CEO, Peter Ammersdorf. And with that, I am actually already finished. I would like to thank you for your attention. I hope you did learn about how HTCR-Services can support you to advance medical development and clinical trials. As I said, please feel free to contact me or my colleagues, and also save the date. May next year, join our symposium in Munich, where we also celebrate the foundation's 25th year anniversary. And with that said, I'm now very happy to answer any questions that you might have.
Yeah, thank you very much for the presentation, and in general, congratulations to all the work of HTCR and the functionality of this biobank. We had a project with the Spitzencluster here to extend that biobanking here in Munich to all clinics, and try to get access to all the different biobanks and pathologies. And I can say from my own experience that it's so much work, so many steps, so many different people groups to be involved, all the permissions and all the things. It sounds always very easy in theory. Well, of course, there are surgeries, there's tissue, there's blood just collected and provided for the R&D in companies, but it's really very difficult to set up an excellent process and to control the quality until it gets to the final laboratory where it's used. So it's really a big effort to do that. And the financing, of course, because there's a lot of money you need beforehand to pay the laboratory, your staff, collecting the tissues and so on until you get at the end something back.
It's a challenge.
Yes. So therefore, thank you for having you here and having that option, the possibility to get bio specimens. Maybe you can add some more details to the symposium you want to have next year in May. It's probably not only celebration, but
it will be not only celebration, we will have a reception in the evenings, but the symposium itself will be solely scientific based. We will have international guests and speakers there, speaking in 4 different categories. Everything is related to the topic, well, similar as this session on research on human tissue. We will have talks on biobanking. We will have talks on organoid technologies, but also we will get deeper into the topic of the ethics behind that and the legal regulatories behind that. And that is what I'm actually most looking forward to, because we will have speakers from different perspectives, different backgrounds, showing their opinion on those things. We are still lacking, unfortunately, a governmental or even EU-wide legal framework for biobanking. So this is another challenge of that, and I hope that we can shed light on that, because we will also have many hopefully a little controversial discussion rounds in each session.
Thank you. It's free to attend, or
the symposium itself is free to attend. We will soon open the registration online. The reception in the evenings, there will be a fee of 50 euros, most likely around about that, so not that much, but to also cover for the catering expenses, which are surprisingly high.
And the registration and everything else I suppose can be found on your homepage later on.
Exactly.
Thank you. So what else? Well, you told us a little bit about how to apply for for tissue for cells. Is there a limit for companies to get biospecimens in quantity, or how often can they get biosamples?
We're basically limited through time and through the number of surgeries that are done. So that is our main influence. If there were 100 of surgeries, we would probably reach our personal capacity. But besides that, there's our archive that limits the amount of samples, but we do not limit the amount of samples that go to one or another partner. We only need to stick to the foundation's guidelines to always have more allocations into the academic sector and less into the commercial scientific partners. But that is basically the only limitation there.
And another question, is it possible to provide the tissue outside of Germany?
Yes, absolutely. We do currently ship on a regular basis to other countries such as Austria, but also the Netherlands, but also Switzerland. So even outside of the European Union, which has additional regulations, but yes, we can do that absolutely.
But are there any limitations on the international level? I mean, because we still have the issue of identification, genetic data, and so on. So, could it be any country in the world, or are there restrictions concerning this data protection thing we have?
So currently we on this security aspect, I assume we would not ship to embargo countries, to embargo states. But besides that, as far as I know,
if the contracts are made and
yes,
everything is clear from that,
exactly.
then you would
exactly. That's why we have everything basically approved twice, not only from an external ethics council, but also internally and also from the foundation.
Thank you very much. We see you at the end again.
Thank you.
And come now to our second presentation from Fraunhofer. The presentation will be given by Matthias Struck, Head of Center for Sensor Technology and Digital Medicine at Fraunhofer IES in Erlangen. Matthias studied computer science with a focus on informatics and medicine in Nuremberg and was research fellow in the image processing and medical engineering department and is now at Fraunhofer for more than 18 years. He should know about novel smart health technologies which are developed and validated in a clinical environment. And his title, you can see that here, quite long, Insights into the Competencies in Medical Engineering, focusing on the validation of technologies and clinical applications. Please, we're curious about your talk.
Thank you for your kind introduction, Mr. Berghammer. As you already mentioned, my name is Matthias Struck and I'm the Deputy Head of our Digital Health and Analysis Department at Fraunhofer IIS in Erlangen. First of all, I would like to thank all of you for choosing to spend this time with me and my slides rather than enjoying a delicious lunch. I'm just kidding. Of course, I'm assuming that's exactly what you have planned right after my presentation, right? So let's not waste any time. I'm pleased to give you some insights into the competencies and technologies within our medical department. And in doing so, I would like to focus on validation aspects as well as on concrete clinical applications. Please allow me to present a brief overview slide of our institute before I dive directly into the hopefully exciting content. The Fraunhofer Institute for Integrated Circuits is the biggest institute of the so-called Fraunhofer Gesellschaft, Europe's leading organization for applied research. And when I speak of applied research in the following, I always mean that we are rarely explore new measurement principles and methods, but rather advance the state of the art and transfer it into concrete industrial applications. Of course, that's the mission of Fraunhofer. And the reason we have been able to grow so rapidly in recent decades is, not at least, due to the well-known development of our MP3 format in the 1980s. And due to our legal form as a registered association, we do not have our own products. Instead, we license technologies and contribute our expertise to research and development projects. Okay, let's take a closer look at promising research and development topics now. I'm personally convinced that medical sensor systems will play an increasingly important role in our healthcare system. What do I mean by medical sensor systems? I mean sensors that are seamlessly integrated into our activities of daily living and that can capture meaningful vital parameters. I emphasize the word can. Why? Because personally, I don't like being monitored around the clock, but I would be the first to use such a sensor in case of a suspected illness, because the data can be collected continuously and without a bias, making it particularly meaningful. I think the topic of medical data analytics with artificial intelligence is self-explanatory and is currently widely discussed. What is important for me here is the understanding that our research, it is not about replacing the doctor, but rather providing the best possible support for the physicians. Later on, I will have a detailed slide on digital patient management. So please let me jump directly to the topic of digital health pathways. In this field, there are two key aspects. First, the question of how can we integrate novel technologies into clinical processes. Even the best technical solutions will not succeed if I have not considered important aspects of usability, for example, or communication interfaces. Secondly, we are researching and validating novel digital biomarkers, such as specific parameters of the human movement system, to, for example, detect Parkinson's disease at an early stage. When trying to obtain a comprehensive view of a person and their health, one should not only focus on vital parameters, such as an ECG and the gait parameters just discussed, but also incorporate as many other modalities as possible, including human emotions like sadness or joy, captured through a camera. With these possibilities of multimodal data collection and analysis, it's naturally possible to conduct many exciting studies. For example, for the automotive industry, in the field of workplace health prevention, or in the therapy of mental illnesses. In the field of comprehensible AI, we are working to develop and improve methods that provide an explanation component for the diagnostic support system, helping to clarify the decision-making process of an artificial neural network. Especially in medical applications, we don't want to rely on a black box, right? And the last research area, which I won't go into further detail on, is medical imaging. On this slide, you can see an example of integrating sensors into daily life, and in this case, into a textile that can be comfortably worn like an undershirt and that can record a 3-channel ECG in medical quality. This prototype solution can be an alternative to the well-known Holter ECG, which is often equipped with many cables and that can be disruptive in daily life. Another advantage is that the integrated sensors are made of dry electrodes, so they don't cause skin irritation. A specific application could be to detect arrhythmias, such as atrial fibrillation, which affects about 2% of the population. On this slide, you can see an example. Sorry, I jumped over one. To develop such solutions, as you saw on the previous slide, both the interaction between hardware and textiles and the seamless integration of hardware and software are essential. And therefore, we have invested a lot of research, not only to achieve high accuracy of artificial intelligence in detecting arrhythmias, for example, but also to implement it as energy efficiently as possible to ensure long battery life. And the challenge here is to shrink a deep neural network in such a way that it can be executed as resource efficiently as possible without compromising accuracy. I think it has become clear to all of you that the multi-model approach addresses many different markets, far beyond just medical technology. And here you can see, without a doubt, an incomplete overview. I've already mentioned the automotive industry. The typical question here is, how can I monitor the driver's attention or detect when the driver is distracted? With the topics of market research, human-centered design, and user interface, the focus is on examining how different target groups respond to new services or how intuitive the use of the new technology is. The gaming industry aims to offer highly individualized difficulty levels so that no player feels under or overwhelmed, but rather remains in a state of flow. And in the following slides, I would now like to dive deeper into the topics of requirements for clinical data collection, general process of typical validation studies for medical devices, and finally, specific clinical applications. Let's begin with the important topic of proper data collection. Artificial intelligence is a hot topic right now. Everyone wants to analyze data. A lot of time, energy, and money is being invested in this area. What, at least from my perspective, often gets overlooked is the understanding that my results, even with the latest AI methods, will only be as good as my data. And therefore, it's worth taking the time to ask the right questions before starting my data collection. Please think back to the prototype for textile-based ECG monitoring that I introduced to you. If the objective is to evaluate the implemented algorithm for arrhythmia detection, I obviously can't use the MIT reference database as a data source, since they were built with a clinical approved ECG device and the signal properties are entirely different. So therefore, I need to initiate my own study with a new prototype. But here come the next challenges. How do I access the corresponding patients? Which arrhythmia do I specifically want to detect? There are many different types. And even if I only want to detect a specific arrhythmia, my patient cohort still needs to include all arrhythmias according to their statistical distribution in order to evaluate my algorithm validly. So let's imagine that we have succeeded in all of this. We now need to have these data sets manually annotated by a designated expert. Because on one hand, less experienced doctors may find it difficult to distinguish certain arrhythmias. And on the other hand, I cannot rely on another algorithm to assist, as it also doesn't provide a clinical reference. And only at this point now can the in-depth analysis of my data sets and the evaluation of my algorithm commence. To collect the required diverse patient data for the validation of novel technologies, we have set up and equipped various specialized laboratories with clinical reference devices at our Center for Sensors and Digital Medicine located at the University Hospital in Erlangen. And this allows us to directly compare the results of the study with the clinical gold standard. And of course, we benefit from the opportunity to directly engage with clinical experts from various disciplines on site. To make data collection as comfortable as possible for all stakeholders, we have developed our own technology for digital patient management, which we call DPM research. And with this, certain parameters, for example, blood pressure, can be easily recorded in the home environment. And as we have already learned, this provides high significance. And the solution we've proposed is entirely web-based and, of course, fully compliant with the General Data Protection Regulation. To plan and successfully complete a study for ourselves or our clients, the following project phases have been proven to be effective. We usually start with a workshop to fully understand the technical requirements and the future business model. Next, we begin off all preparatory measures, such as defining the exact endpoints of the study, biostatistics, patient insurance, and the ethics application. Afterwards, patients can be recruited, the measurement system can be set up, and finally, the actual data collection can begin. If needed, we can then assist interested parties in optimizing their technology as well as in the clinical approval of their medical products, although I won't go into detail on that today. For all studies that go beyond technical validation, but also addresses clinical efficacy, we of course comply with the medical device regulation and consult with both internal and external legal experts. Now, I would like to take a step back and provide an overview of the opportunities Fraunhofer has for commercializing its own technologies and expertise before I conclude my presenting with concrete clinical applications with our partners. The first opportunity is that we solve a specific customer problem or provide them with measurable added value. Illustrative examples include optimizing an algorithm's classification accuracy using the latest artificial intelligence methods, or implementing a data interface for a medical device. So pretty forward. Secondly, the development of new business models with technologies. As you already heard, for digital patient management, for instance, we have developed a concept based on our DPM research which allows certain studies to be conducted entirely digitally. So therefore, no one needs to come to a laboratory, which is quite comfortable. And finally, particularly important to Fraunhofer is always the technology transfer. The second project example is the non-invasive determination of pressure in the human brain. For this, we have been collaborating very successfully for many years with the company Sonovum from Leipzig, which has a device for ultrasound-based recording of the so-called brain pulse curves. And based on the brain pulse curve, we have developed an AI-based algorithm that can differentiate between normal and increased intracranial pressure. And clinically, this has a great advantage that many patients with traumatic brain injuries or stroke patients may be spared the need for an invasive probe in the future. And we are about to begin the first measurements on stroke patients at the clinics at the University Hospital Erlangen. The next project example comes from the psychosomatic field. As you all know, healthcare workers, especially during the COVID-19 pandemic, were subjected to immense physical and physiological stress. In Germany, around 1 in 3 healthcare workers quit their job during the pandemic. And in Europe, 9 out of 10 healthcare workers considered it. Of course, this is a catastrophe for all healthcare systems, or for our healthcare system. And together with the University Hospital in Erlangen, we have set up a study with healthcare workers to detect overloads as early as possible. Alongside psychosomatic questionnaires for self-assessing life satisfaction and measuring cortisol in the blood as a reference for stress, we recorded heart rate variability using a variable very similar to the one I introduced to you at the beginning. And we found out that the self-assessments from the questionnaires often did not align with the physiological data. For example, some individuals felt they had gained a bit of distance from work after or over the weekend, but this feeling was not reflected in the data. And such discrepancies can eventually lead to burnout. Allow me to present one last project example from robotics. Children with impaired socio-emotional abilities, for example, autistic children, often not only have difficulty expressing their own emotions, but also struggle to correctly interpret the emotions of those around them. And this can lead to socially challenging situations. Imagine a child who suddenly starts laughing in the classroom while the teacher is discussing a train accident, for example. Since we know that autistic children are often highly intelligent and technology-oriented, our idea was to have them learn socially appropriate behavior in a playful way using a robot. And together with Humboldt University in Berlin and the other parties listed on the slide, we have achieved remarkable results here. With this slide, I would like to sincerely thank you for your attention and hope that I was able to provide you with some exciting insights into our daily work in the medical department at Fraunhofer IIS. Thank you very much.
Yeah, thank you very much for the interesting examples. It's really impressive how the world is changing year by year in terms of technologies and how to apply them at the end to life sciences or to medicine. So I think for our companies in the network, it's that aspect of collecting data within clinical trials is probably one of the most interesting applications to get non-biased data, as you said in the last examples, what the patients feel or write in forms. Does not reflect necessarily what is really the biologic within the body. So that's a thing which we wanted to bring here with this lunch talk, that there are technologies to help and to do that as well in a comfortable way for the patients, because if they don't accept it, then whatever technology is not helpful because it doesn't work. Concerning this ECG, is the textile for capturing an ECG already commercially available, or is it just a prototype you have?
Yes, also in the form I presented to you today, the textile is not yet commercially available. However, we did have an earlier version, not as a medical device, but as a lifestyle product, which was marketed for a few years by the company biotech, and it was for sports applications.
And that let's say it in another way. The results from that textile from the ECG there, is it comparable to the ones I get in the clinic? Well, you say you said already the signals, of course, are a bit different, and you cannot compare it in the same way. But let's say in the meaning of what is a malfunction or something which is not in the perfect range, that's comparable.
It is comparable. We have clinical quality, and we conducted an own study with the clinics for cardiology in Erlangen, and we had results comparable with those of clinical validated ECG Holter systems.
Because we have now, of course, the more and more evolved smart watches and so on, and they say that they can analyze ECGs or do ECGs as well and get results. But that's a different, or would you say that this is already coming close to the medical application?
Apple is a great product, no doubt about it. But Apple does not provide a continuous ECG. In the latest Apple Watch, for example, maybe you will wear it at the left side of your hand. You have to put the finger from the right side to close the circle to the heart. Just letting the watch measure on the left, on your left hand side, does provide an estimation of the pulse, but it's not the same as in real ECG. So it's somehow difficult to complain it with the end user. I completely agree, but it's a different complete wording of quality here.
And the other question, your DPM research, that's a platform to, as far as I understood it, to collect the data in clinical studies and to have the data protection and everything included to easily use it in then officially official clinical trials.
Exactly.
What does a company has to do if they want to use it, or how do you come together?
Yes. Now, first, we naturally need to understand the precise technical and non-technical requirements for the planned study. And so this will show us which requirements we already covered by our technology and which maybe not. And to address any gaps, we could add the necessary effort as part of an R&D project. And for the duration of the study, a licensing agreement would be established for the use of our technology.
Okay, so you would pay a fee for the license using that within the clinical trial.
Exactly.
But if something is missing and has to be developed, that could be a kind of project within that usage.
It's usually a small project, 1 to 3 months exactly. And we will fix the gap and then go into the study.
And one thing is data protection, of course, ethics, permissions, and so on. But the other thing is safety of data. How is that included into your platform?
As shown on my slide about BioM research, our technologies comply with the general data protection regulation. So whenever personal data is involved, and that's mainly the case in clinical studies. So we therefore implemented appropriate mechanism for data encryption and of course also for authentication. So standard computer science stuff with the state of the art here.
Okay. So thank you again. I would like to have Mareike again back on the virtual stage. Do you have questions to each other?
So I can maybe start with one. I really enjoyed the example that you showed us, Matthias, on the voice study. If I recall correctly, that was the one dealing with, well, predicting burnouts, right? Yes. Like kind of. So I'm not really into the diagnosis criteria, but I know that in most psychological disorders, those are only based on questionnaires. So that is quite a controversy that you are starting there to say that the questionnaires do not reflect. Yeah. Well, the maybe actual patient situation or possible patient situation. Can you elaborate on that?
Yeah, so what was pretty amazing for us is to see the questionnaires and you see a sentence like, yes, I feel relaxed Sunday evening before watching in Germany Tatort, for example, yes, but the data doesn't show it. So subjective feeling and objective data does not align. And that's crucial. So when you do that for 2 or 3 months, that may lead to burnout or something else. So you can go to the person and say, hey, data says something different. Maybe something is wrong with you.
Or with the environment.
Exactly. Yeah, exactly.
Thank you.
What else do we have? It's to Matthias. You having clinical trials or especially for new medical equipment is a kind of a almost of a co-production between Fraunhofer and the company, leading to a result in the end and the trial in hopefully in a positive result. But do you have also options for further helping the company in the development of the business model?
Yes, definitely, if needed. As this is very crucial aspect of the later commercialization success, for sure. So our employees from the business development department are highly skilled in the development and validation of sustainable business models. That's some standard work package joining industrial corporations, right?
And can you say something about the cost of having that clinical study with you?
It's a difficult one. It always depends, for sure. So let's start with a workshop and see where we stand. It's completely different. We had small studies for, let's say, above 100,000, but of course, clinical studies can reach over 1 million. That's no problem. So somewhat in between, I would say.
Okay, thank you. Mareike, we're talking a lot about collecting biosamples, having the processes and so on. What about the patient? What do the patients have to do in your, well, they are the most important ones.
They are. We totally rely on those. They basically don't have to do anything. Like we provide the framework, and they don't have to go through any additional procedures. Everything that we practically do is within the frame of their surgery, so it does not affect the surgery itself or the time or whatever. The only thing that we ask the patient for is their consent. And that is not done by us randomly, but by the physicians that are also doing the informed consent for the surgery itself, for the anesthesia and all that. So that is just like one more sheet to read, to understand, and to sign. And the patient is always free to say no, and also to withdraw that consent afterwards. But we really try to make sure that the patients themselves don't need to have any additional tasks, don't have any effort on that. So to leave them to their recovery, basically.
Thank you. I think we are done. I don't have here open questions. We are a bit 2 minutes over 1 o'clock. So I would like to say many thanks to the speakers for their interesting insights. And many thanks to you all for listening and watching. And I hope you will be back next time or on other occasions. See you again. Thank you very much.
Thank you.